Thank you very much. It's a pleasure to be here, and I am a general surgeon. I operate mostly on the intestinal tract, the stomach, the gallbladder, the pancreas, in that region. So I'm going to talk to you a little bit about some of the work that we've done over the last 20 years to try to contextualize it to the idea of applying precision medicine. Now, we're lucky at the University of Chicago that we have a lot of centers that interact with one another, just like the microbiome. Central to that is Jack Gilbert's laboratory and effort. We specifically recruited him to the Department of Surgery because there are a lot of problems to solve there. So there's a picture of Jack. You've just heard him speak, and I always put this up because what is microbiome medicine? We actually are talking about microbiome medicine, and it's the application of the unaccounted for dimension in human biology to patient wellness and disease management. And so, when you look at that dynamic microbial consortia, and you start to think, especially in view of Jack's talk, everything that goes in a patient's mouth gets processed by the microbiome, and metabolites are released. I mean, that's daunting. I mean, the very vegetables that you eat in the environment from which those vegetables came and how they were sprayed and treated and grown and what kind of dirt, to the very metabolites that the bacteria are going to release in response to those vegetables, or how that meat was processed, or whether those animals got antibiotics. This is a daunting sort of dimensionality to that, all of that. And one thing that's been very interesting is that your microbiota release metabolites that literally directly interact with your DNA through acylation and methylation events. They're metabolites that literally, they don't change the structural genes, but they change the way gene expression develops in an individual over the course of time and perturbations. And everything underneath is affected. So I always quote Borges, everything touches everything is not a big stretch when you think about it. So this is an article that one of our residents wrote when he spent 3 years at the American College of Surgeons right here downtown and studied why patients end up back in the hospital. So one of the big metrics, the surrogate proxy markers now for if you're a good surgeon, is how often do your patients come back to the hospital, your readmission rate. And it's because that's the most costly thing to happen to the patient. Fix their hip, take out their gallbladder, you have a big bounce back rate. You're sort of penalized for that for all stakeholders. Your department chair's mad at you, the insurance company's mad at you, the patient's mad at you, the family's mad at you, everybody's mad at you. And sometimes that's preventable and sometimes it's not. And as you might imagine, there are two reasons as a GI surgeon that people come back. One is an ileus, which means their stomach blows up, it just doesn't move, it stops working. And the other is an infection. And any intestinal surgeon would say, oh yeah, those are the two. Why did this person spend all this time writing this paper? It's so obvious. But he documented it in over 14,000 records. So if you ask any general surgeon, what causes an ileus, he or she will come up with, a bunch of explanations that really have no scientific basis to them. And that's what we call dogma. If you ask a surgeon who are the three best surgeons in the world, they'll have trouble naming the other two. Okay, so that's one problem that we have. So it turns out that this postoperative ileus, this paralysis, this lack of function of the bowel, starts right here where bacteria adhere to the epithelial surface. They change their phenotype, they affect the cells below, the dendritic cells, the antigen-presenting cells. They communicate to the macrophages, and those macrophages are in direct contact with the nerve endings in the bowel. I mean, so when I say everything touches everything, it kind of rings true, doesn't it? Sort of what you fed the patient, what antibiotics they got, what hospital pathogens they acquired while they were in the hospital, when they went home, did they take narcotics, et cetera. And you get this, what we call a paralytic ileus, and that's one of the big reasons it bounced back. I can't poop, I'm all distended, what happened? And in the center of this drama lies the macrophage, which is the main protagonist here. And if I tell surgeons that ileus is a postoperative inflammatory disorder initiated and driven by an altered microbiome, they'll look at me like I have two heads. But our internal medicine colleagues have actually done some very nice work in this area showing this. So I just want to run through a few ideas with you about how disabling surgical complications are unknown and not really reconciled at the level of molecular mechanisms, because we don't really know what's in the gut at any given time in a given patient. I mean, think about this. We're operating on older, sicker patients. Everyone takes a Z-Pak. If I walk into my patients in the preoperative area and I say, have you taken any antibiotics in the last 6 months? 50% or more will say yes. Well, I take a Z-Pak. I get urinary tract infections. I take Cipro. I have diverticulitis. My doctor gave me a pile of prescriptions. I just fill them out and take, They're all taking antibiotics. And so what am I starting with? So now we need to sequence, if we really want to know the answers, because culture is inadequate, we need to sequence at the 16S rRNA level or deeper and find out not just what members are there, but what community structure, what relative abundance are they to each other. But that's not enough. We need to know their function, what genes are turned on, what proteins and metabolites are being expressed. And then if you want to ask this question, and this is very region and spatial specific, does this community express the human phenotype, as Jack mentioned? You actually have to colonize germ-free mice with the actual segment of bowel that has the ileus. You got to get that sample. Or in an anastomotic leak, you got to pick that area out, and you have to put it back into a mouse model to show it. And I think the most striking recent finding for me anyway is Parkinson's disease, which is, as you know, a disabling condition of the neurologic system. And if you literally take the feces from a patient with Parkinson's disease and put it into a mouse that's raised germ-free, it will develop neurologic symptoms similar to a Parkinson's disease patient. And there's something that if you ask a neurologist, is Parkinson's an infectious disease? They go, nah, we biopsied the brain 100 times. There's nothing growing there. But everything touches everything, right? Think about the layers, the dimensionality of this problem. And think of what I'm showing you here, how complex it is to measure all this stuff and then manage and display that data meaningfully. So this is what you might think of as an infection, but I want you to rethink of the definition of an infection. It's the perturbation in host function that occurs as a result of a complex interconnectivity between various species of microbes, i.e. a community and host physiology. And that system's interconnectivity is very much activated, perturbed, disturbed, messed up, as my daughters will say, when you operate on patients. You give them, think about this. Patient comes into the hospital, they're starved overnight, you operate on them, you give them blood, you give them antibiotics, and most of them go home. And it's because these bacteria in the gut intercept host signals. They talk to one another through quorum sensing, and there's not only interkingdom bidirectional exchange, but there's intraspecies and interspecies exchange. So you have this bacterial social network, and these emergent bacterial phenotypes or properties are actually unique to the environmental context, and I can't emphasize that enough. So as my colleague James Shapiro, who's a molecular geneticist at University of Chicago, says, there are no units, there's just interactive systems. So here is what this looks like when a surgeon operates on you. You have this engagement with the host in your gut microbiome with all these different cellular elements within the host tissues at the level of the wound as well as the level in the gut, lung. And you have this iterative loop of reverse causality. But here's the cool thing. At the end of all this, 98% or more of the time, there's molecular detente. It's in the best interest of both sides, both organisms, to leave each other alone, right? You can instantiate game theory into this. But every now and then, and if you just think of all the elements in here, like when you're flying a plane at 25,000 feet and you get turbulence and it's unanticipated, pilots have to respond, bacteria have to respond, and these infections, these complex problems that have eluded clinicians develop. So here's 20 years of work from my laboratory using the model organism Pseudomonas aeruginosa, where we identified the different host elements that are released, how they interact with specific elements within bacteria, in this case Pseudomonas aeruginosa. And look at that, opioids. Think about that for a minute. You know, there's a war against opioids from the White House all the way down in this country. because people are getting addicted to them, and many people die of an infection. But we found that opioids can directly bind to this multiple virulence factor regulator in Pseudomonas. It's a transcriptional regulator, both exogenous and endogenously released opioids, and activate its virulence. But what was really interesting, and Jack has done some of this work when he worked on the microbiome of the West English Channel, is that under low phosphate conditions, that Pseudomonas aeruginosa really gets revved up in terms of its virulence because of a phosphate sensing protein that connects to the quorum sensing signaling system. But under high phosphate conditions, you inhibit this and you never get any virulence expression. So one way of interpreting that highly reductionist experiment is to say when bacteria get the nutrients they need, they become insensate to these incoming host activation cues. And that's why Sam Miller wrote this paper. He said, are bacteria just looking for food? Because there's a direct connection between these membrane sensors and how bacteria are actively induced to express pathogenesis against their host. They really want to leave us alone. They want to live in symbiosis. But I mean, just think about what we do these days. We take antibiotics, we get on a plane, we fly to China, we come back 2 days later, we bring viruses with us, we eat food there, we bring food here. February in Chicago, you eat plums from Chile, you drink wine, you get sleep deprived, and you're like, what happened? Why don't I feel good? Okay? So the American College of Surgeons really has been trying to understand how to make surgery safer through what we call these enhanced recovery programs. And if you look at these programs, one of the major pillars of these programs is stop starving your patients. Feed them right away. Just think of that in the context of what I just showed you. And here is the enhanced recovery after, the basic tenets, and this ERAS program came from Europe. Like their big thing is feed right away and don't use any morphine or opioids. And their patients recover almost faster than ours do because we tend to use a lot of opioids in this country. And then we prepare the bowel for surgery in a way, and we all have disagreed on it. And the cool thing about this is there's no science behind this. We've been using the same procedure for 50 years. And if you ask a surgeon, especially one my age, they'll say, listen, Sonny, I've been doing this for 50 years. I haven't had a problem. He's had a problem. And most of our surgeons now are she will have a problem if they follow this. So here is a very nice study by Fiona Guanet. We do these bundles. We sort of do stuff to people, and we call it evidence-based medicine. It's not evidence-based, and there's no science to the evidence. We do stuff. Things go down like this. We give a mechanical bowel prep, drink this stuff, clean out your bowels, take some antibiotics. We're going to give you lots of oxygen. That's good. And hey, look at this. We reduced the rates by 50%. Isn't that awesome? And I say, hey, what led to that reduction? They go, well, look at what we did. We got the glucose control and everything. And then if I say, oh, the bundle did that. Excuse me, why did these patients develop infections? And this is where the hand-waving, use of metaphors, and blaming of nurses and anesthesia comes in, okay? Well, nothing's perfect. I got the damn nurse, the doctor. This is wrong. You want to get, is this calibrated to the patient's microbiome? It is not. We give the same antibiotics we've been given for 60 years. How is optimal fine-tuned? Too much oxygen can be bad because you release oxygen radicals. Is that good enough? Is that good enough for your loved ones? Do you want your surgeon to tell you you've got a 7% chance of infection, but hey, don't complain, it used to be 14%. I want it to be 0, and I want to know why you went from 14 to 7. So think about this. Everything I showed you in that schema kind of fits into that pattern, but there's no precision medicine to it. It is empiricism ad absurdum. So let's talk about this problem of an anastomotic leak, which Jack and I feel that we've solved. Now, you may look at this and say, when a surgeon sews all that back together, if it leaks, that's a crappy surgeon. I mean, in fact, in our morbidity and mortality conferences, everybody gets up when one of these leaks, and it's very rare, maybe 3-4% of the time, And they start talking, well, did you use 2.0 or 3.0? How'd you, with the stapler, they're talking about stuff that has no evidence to it in terms of causality to the leak. So we frame this as an infectious disorder because surgeons I know who I'd let operate on myself and any member of my family who are outstanding still have these leaks. Why is that? Well, they had a bad day. No, they do it the same every time. And still, the best surgeons across the entire globe have these problems. And it's eluded them, and they keep saying the same thing. So two people suffer when a leak occurs, the patient and the surgeon. You feel it's your fault. You've been told you didn't stitch it right. It's not true. So Ben Shogan, who worked in my laboratory, did a very nice study and just looked at, as Jack showed you, opening the bowel, and showed that there was a Bloomin' enterococcus in that model using 16S rRNA analyses. And we did some phenotype and proteomics also. And he found that E. faecalis can actually become predominant in the population because you open that anastomosis to air and kill all the obligate anaerobes. And when it grows, it actually gets cued, it gets activated by host signals and makes collagenase that actually is a flesh eater. It turns from a commensal non-flesh eater to a flesh eater. And he went through all the details of how it activates MMP9, which is a host protease, breaks down collagen 1 and collagen 4, and really did a beautiful job of showing for the first time ever the molecular basis of an anastomotic leak modeled as an infectious disease. And then Bader Shakshir, who's now a pediatric surgery fellow at WashU, worked in my lab for 2 years, essentially showed the same thing when he put little morphine pellets in mice, slow release morphine, like what would happen in the hospital or when your doctor would give you some morphine to go home with or some codeine. And he found that you get virulence expression in the enterococcal species that predominate in the form of enhanced collagenase production and leakages. And now we've started collecting these organisms. We have Pseudomonas, Serratia, Enterococcus, and Bacillus pseudolus, all that make very high collagenase only under the right environmental context. And you have to model that very importantly. The context is important. To make this happen in mice, we have to make them NPO nothing by mouth. We starve them overnight like you would a patient. Then we give them antibiotics, kill all the good guys, and then we do a little tissue injury, like Jack showed, to cue them up. And then when they're exposed to these organisms, bingo, they chew right through the anastomosis. But it's proper modeling. Now I'm going to finish up in the last few minutes that I have with surgical site infections. So the worst thing that can happen to you if you have your hip done, your back operated on, your spine, anything, is to get a wound infection, because it can be disabling. If it's like in your spine, you could be on antibiotics for 6 months and bedridden or in a wheelchair the rest of your life. What causes a wound infection? What do you mean, Alverdy, what causes a wound infection? Somebody dropped something in there. What are you, stupid? Of course, you open the wound, somebody contaminated it, they didn't realize they did it, and you got a wound infection. Unproven. Now, if you did wound cultures at the end of an operation, so you do 1,000 of them, is it surprising that there's no correlation between what is growing there at the end and who gets a wound infection? Now, our infectious disease colleagues tell us we have to wash our hands, be sterile. I'm like, 15 years of formal education and that's all you got, wash your hands? What do you think, I'm not gonna wash my hands? Watch this, and do this, HEPA filters, reverse this. I'm like, that's not it. We still have infections. How can we have infections? Well, you're not being careful. You're not washing your hands. The best hospitals in America still have infections. In fact, if you could stand up and say, University of Illinois has a lower infection rate than the University of Chicago, patients would flock here. They would flock here. If Rush could say they were better than you, they could market that. It'd be on TV every 5 minutes. They can't. We all have the same infection rates. Why is that? Same as Hopkins, same as Duke, same as UCSF. So when you do a perfectly clean case, nothing grows. When you do a complicated case, like if somebody perforates or an ulcer blows or something, it's contaminated, and yeah, they have a higher probability of infection. It's not deterministic, it's probabilistic. But what about everybody in between? Everybody in between makes no sense. There's several orthopedic studies where they culture it, and it's positive. Positive. And you go, oh, that patient's going to have a terrible wound infection. They don't get wound infections. Why is that? Where do these pathogens come from? It's a mystery. Who's doing any work on this? Nobody. Okay, so here's Monica Kreselek. She worked in my lab for 2 years. She's going to be a colorectal surgeon. Next year, she's going to the Mayo Clinic. And she did this study, and we modeled what's called the Trojan horse hypothesis. We hypothesized, and this has been hypothesized before but not proven, that under certain circumstances, intestinal bacteria like MRSA, methicillin-resistant Staphylococcus aureus will colonize the gut or other mucosal surfaces. It will get cued somehow. It will be taken up by a neutrophil. That neutrophil will end up in the bloodstream where there's molecular détente between the neutrophil and the bacteria, and it will seed the wound. How are you going to show that in a patient? That's going to be really hard. So she did this the right way. She modeled this perfectly, gave the animals antibiotics, starved them like you're supposed to, like we do with our patients, gavage down, meant orally put down some MRSA that was labeled both GFP and bioluminescent strains, then caused some trauma to the wound, then created a big surgical stress by taking a little piece of the liver out. Now this animal looks like a human that we've operated on. And she could show you actually get a staphylococcal wound infection when you do that in 10% of the mice. Not 100%, 10%. And she gated the neutrophils out with flow cytometry and could actually see that the neutrophils, they fluoresced nicely, like we hoped they would, and they were inside these neutrophils alive. Now, two things happen when a neutrophil eats a bacteria. The bacteria kills a neutrophil and then kills you. Or the neutrophil releases oxygen radicals and kills the bacteria, right? That's how it works. Makes bleach, makes all other goodies in there. But these things are living together, and there's molecular détente. Turns out they're small colony variants, but they're really rare. Okay? And then what she did was show that the MRSA that she gavaged down persisted in the gut, and that same MRSA inside the neutrophil was the one that ended up by doing deep sequencing in the wound. This is the first evidence, and it's a low incidence event that this could happen. But what I want you to understand is there are multiple contingencies that must be fulfilled. First of all, the competing flora must be disrupted so MRSA persists. If you have a normal gut flora, I mean, I don't have MRSA. I've been actually tested. I've been a practicing surgeon for 25 years at a university hospital. Why am I not colonized? Well, I've probably taken antibiotics once in the last 20 years. You just can't, my gut microbiome has home field advantage. It will not let anything in, okay? But think of your patient that took a Z-Pak that had a virus 3 weeks before you operated on them and went and visited their aunt or grandma in a nursing home and gave her a nice big fat kiss while they were on antibiotics, and now they're colonized with MRSA. They're healthy, but they have MRSA in there. The second thing is the MRSA and the neutrophils have to achieve molecular détente. Most of the time, your healthy neutrophils are going to clear that MRSA if it ends up inside. Now that MRSA has to home to the tissue injured, so the surgeon has to do not just a 20-minute operation, but maybe a 4 or 5 hour operation. And then the tissues have to be receptive to MRSA, and this is what we call a rare adverse event. But when that MRSA, let's say in 3% of patients comes up, you know what the hospital epidemiologists do? They start culturing the room and the patient and the personnel, and they go, somebody dropped it in there. I'm like, excuse me, we dropped it in 200 patients beforehand that didn't get it. Well, we're not interested in that. We're just interested in the one that got it, because we're detectives. We don't want to know when murders didn't occur. We want to only investigate when the murder occurred, okay? So here's where we need microbiome medicine. I only have one more slide left after this. We need microbiome medicine because this is what surgeons are doing now. They said, well, MRSA is a problem, gets in that wound. I got an idea. Vancomycin powder. We'll open the wound up and we'll powder it up, and it'll last in there 3-4 days. That's going to be a good idea. So this group looked at 1,200 consecutive surgical cases, spine surgery with vancomycin powder. They found 2.83%, which is actually high. Spine surgery should be about 1.5%. And let me just tell you something. If you have your spine operated on and you get a MRSA infection, you might be in a wheelchair the rest of your life. That's not a joke, 2.83%. And here's what they found. Of the infections that developed in that group of 1,200 patients, the 34, look what grew. And some of them were polymicrobial. So you'd say, wait a minute, you killed one, and how did these other ones get there? All right, somebody spit in the wound. Somebody put poop in the wound. Call the epidemiologist. They'll come in there. They'll take everyone's fingernails and find it. It's wrong. Where did they come from? We don't know. So these programs need to be analyzed at a very high resolution, granular level so we can understand what we're doing. Because this is what we do now, and we're flying by the seat of our pants. Personalized medicine applied at the level of the microbiome will prevent postoperative infections and save lives. Thank you.